Today's speaker is Karen Miga. She's Associate Professor of Biomolecular Engineering, UCSC, also Associate Director of the UCSC Genomics Institute. She co-leads the Telomere to Telomere Consortium that released a fully complete assembly of the human genome in March 2022. Also in 2022, she was named one of Time 100's most influential people. Also in 2022, she participated in the closing plenary session of the Clinton Global Initiative meeting, where she discussed the future of equitable genomics research with former President Bill Clinton, U2 lead singer Bono, and the Director General of the World Health Organization. In 2023, she was named a Searle Scholar to study uncharted heterochromatin regions of the human genome. The Searle Scholar is just not another honor. It's an award made annually to 15 young faculty in biomedical research and chemistry in the U.S. And aside from being a prestigious award, it comes with a healthy stipend. Today, her talk is a little different from what I have. Rebooting the Human Genome, New Variant Discovery in the Era of Complete Genomes. So thank you so much.
Well, I love getting invitations to come to beautiful places like this. So thank you again. It was a joy to come to Arboretum. And I have to admit that Barry's my neighbor. And my mother-in-law came in last night for dinner and she's like, I just bumped into Barry. He's looking forward to your talk tomorrow. And I said, oh, you must be wrong. It's on Friday. So I have to admit, I did have a heads up by the wisest, most valuable person in my family yesterday, so I apologize for being late. But there are definitely 10 students who took the midterm during your lunch that appreciate my presence on campus. So today I'm going to have kind of a broad talk. I have, for the most of my career, focused on the human genome. And I think most of us think about the human genome perhaps as being this biochemical property. We often think of it as double helixes that are kind of kept within every one of our trillions of cells in the body. Sometimes we even think about the Human Genome Project as being this type of book. I'll use this metaphor throughout. And we've been celebrating this book of life as early as the first release of the first Human Genome Project, which I'm highlighting here back in 2000, really showcasing that this was a really foundational celebration that happened. Of course, I like to use this moment of pride for our campus whenever I talk about genomics to a broad audience. It's not always focused day to day on genomics. That the actual spark that launched the Human Genome Project happened here at UC Santa Cruz. My understanding of history is that this was actually a vision that was brought in by the chancellor at the time. And there were funds that were going to be used for the astronomy department. And there was some, I guess, suggestions, maybe using these funds that would originally be used for the launch of some of the Keck Observatory to launch the next Human Genome Project. We brought in a ton of expertise onto our campus at that point, and it is still recognized as being that pivot moment of the launch of the first human genome. Of course, there was a lot of opinions about this effort. It was kind of building as it was going. There was no start to finish plans. Oh, I'm thinking this is the wrong. I'm going. Forgive me. I brought on a talk, but I'll keep going. And so the Human Genome Project, of course, who represented it. I often ask this when I give a talk, so forgive me. I pulled the wrong slide deck. I don't want to pull you guys on this, but sometimes I want to ask, is it a volunteer? Was it 8 individuals or 20 volunteers that were advertising for a newspaper? And I do ask this from my students because they're always surprised to know that it was 20 individuals from a newspaper that were brought in. This is the actual advertisement that was put in the newspaper, actually asking for these volunteers. I work with a lot of historians now that discuss the Human Genome Project, and so this is actually the front page that no one's ever seen before. Very few people have access to it, where they're actually asking for only one man or woman to donate. So I think there was some kind of confusion from front page to back page. And so when we think about who's represented in the Human Genome Project, originally what we're talking about is 70% ended up being one individual. The rest of the Genome Project, roughly 30%, was the remaining 19 different individuals. What I'm trying to show you is the representation of what we would call differences or diversity in our sequences among people. And so you can see I'm breaking these into population descriptors such as African, admixed, American, East Asian, European, and South Asian. And you can start to see the different color blocks that are being represented. This is largely because that individual who is 70% happened to be admixed. So we're seeing a lot of this representation. Of course, in 2000, we celebrated UC Santa Cruz as being the map makers. We had the first release of the Human Assembly from UC Santa Cruz itself. I always like to credit that this was less powerful than your cell phones today. It was really wired together with about 100 Dell workstations. And this is showing a much younger Jim Kent and much younger David Hausler, who many of you know, who happen to be both my postdoctoral mentors. And this is just showing that from UC Santa Cruz's campus, we had this type of explosion of kind of outgoing internet traffic when we released the first human genome from our site. So since that time, we've really come to honor this Human Genome Project through two amazing foundational pieces of publications. This is both in Nature and in Science, and it's led to a fundamental step change in how we understand human biology and disease. As I introduced human genomes as this type of helical structure or biochemical property, I now want you to switch gears and no longer think of it that way. Because since we established that first map, and since we released it on the internet for anyone with a computer to download it, this fundamental resource has now become a coordinate system, much like how we use Google Maps. And no one's looking at Google Maps to look at those kind of precise GPS coordinates, right? You're trying to figure out how to get from the Arboretum up to the library on campus. And we're using these maps now the same way. So all of your chromosomes, position 1 to 10, now form this very critical coordinate system for scientists to operate on. So for the past 20 plus years, billions of dollars have been spent on this coordinate system around the world. So now if I drop a pin on your chromosome position 1001, I know that's a variant that's really important for understanding a particular human trait or a human disease. And the important thing too is this critical tool has been really pushing us ahead in understanding basic science, clinical genetics and genomics, our own human histories. And the way that this works is that it's completely open access. That was by design to ensure that we had the broadest use of this tool. There's no kind of fee for use, or you had to kind of apply to be able to use it. And this had it open up for everyone around the world. The impact was huge. I often like to talk about how it has had an immense property in understanding identifying genes and gene variants for understanding diagnostics as well as therapeutics. And for those of you who think about the economy, which is sadly how we have to communicate science to most people now, I always like to say that this has been the single most influential investment that's been made in modern science. We know that for every $1 of federal spending, it's contributed to the generation of 141 in the economy. And I always like to also say the place I'm getting my notes from data collected back in 2010, 15 years ago. So this is only good. So that was a pretty nice ramp up to give you kind of a historical view of what I'm doing. But now I want to go into what our team and teams even that I work with around the United States, around the world are trying to do now to improve upon this foundational tool. I want you to leave my talk knowing that the original Human Genome Project, the one that's been celebrated for decades, has room for improvement, and that the upgrades that we're working on now will have increased benefit to the public. The first piece of evidence I want to share with you is actually a figure from the original Human Genome paper. And what I'm hoping you can see without me even needing to point to it are these giant red blocks that I'm using to attract your attention. These are the massive gaps that we knew were always present in the first map of the human genome. I love hearing sometimes different metaphors. Some people think about the human genome as being kind of Swiss cheese in terms of having these giant holes of information. There aren't a lot of genes in these particular gaps, which is why they mention it in the paper when it was released. However, they do represent a large fraction, about 8% of your total genome, or roughly 200 million bases that were just completely missing or incorrectly represented. If we just think about the size of those particular holes and we consider them equivalent of blackout zones, I like to think about this because I've already introduced the human genome as being like Google Maps. And so if you're ever on Google Maps and you zoom in to certain places, like I'm showing you here, and you see these like blacked out secret areas of the world, these are what I'm kind of using as a metaphor for blackout zones. If you were to consider the whole surface of our planet, and you were to consider the size of the human genome that's missing from information, you would be thinking about a blackout zone that would be like blacking out the entire continent of Africa, just to give you some idea of how big of a gap we are dealing with in terms of information. So as introduced, thank you very much. Back in 2018, I hosted a pizza party up on E2 on our campus with a good colleague of mine, Adam Philippi. And at that time, I had been working with Dave Deemer and Mark Akinson on some of this really exciting technology that was on campus, known as nanopore technology, where we were starting to see DNA being read on what we call a read that could get up to 100 kilobases in length for the very first time. And we had this vision that perhaps if we generated enough data, we could start to look at a complete map of a human chromosome. A chromosome, as you might see here, has 2 caps on either end with a telomere. These are known biological structures. They're the end of chromosomes. They go through what I'm showing you a primary constriction, or where you sometimes see an X on a chromosome. Those are centromere structures all the way to the end. And we had a hypothesis, could we complete these maps for every human chromosome end to end or telomere to telomere? And so it was from that fun pizza party that we decided to work together and start to generate data. That was back in 2018. We announced the first X chromosome release in 2019, and then we launched a global consortium in the beginning of 2019 into 2020. I don't know if you guys remember, that was a pretty big year. So we walked into a global pandemic with a completely open grassroots effort of scientists around the world who just came together for this one mission of trying to complete the map. Now, we did this because we knew going into it that it wasn't just climbing Mount Everest to complete a map, but there's actually important genome biology that has been missing from a lot of our studies so far. One is I can tell you that the genes that you may be familiar with, sometimes there are multiple genes that look a lot alike. They're called duplicated gene families. So you might have gene A prime, GAA prime, things like that. And having some understanding of these duplicated genes is really important for understanding how our genomes work. Also, having 2 places in our genome that look a lot alike can lead to chromosomes breaking and reforming connections in areas that they should not. And so these are also areas of genome instability that have been tracked for many years. And so we really want to have more complete maps for that region. Also, these primary constricted regions, these are areas of centromeres, which we know are important for proper chromosome segregation and centromere function. That's been my lifelong research project in my own lab. We've never been able to really see what lives in these regions, what contributes to proper chromosome segregation. But we know when that goes wrong, if you have something that errors out in that particular region of the genome, you can have 1 too many chromosomes segregate. And that can lead to all types of different diseases or be associated with early birth defects, with cancers, and even with loss of chromosomes as we age. And then if you think about the acrocentric chromosomes, there are 5 of your chromosomes that have very, very small little arms. However, they're critically important for life as we know it because they harbor these very important tandem repeats that make up our ribosomes, or these important RNA structures that are really critical for translating proteins and making sure that we have the type of biological constitution we need to be to live and to create cells in our body. So why was the map incomplete? If we think about it as my metaphor as a book, once again, if we think about the segmental duplications or these copy number variants, it's not like you're just seeing one word being repeated over and over again. It's almost like you took a whole paragraph from a book and you copied and pasted it identically somewhere else. And that's exactly what I'm trying to show you here. Has been happening in our genome for many years. We have big, large segments that have been copied within the same chromosome or between chromosomes. Now, this is really important because these particular near identical pieces of our DNA, they can be thousands, sometimes a megabase, I think 400 kilobases as I'm linking here. We're now in the scope of looking at hundreds, if not thousands of genomes. So we know that these are quite large. And they contain genes. And there's been a lot of really fun stories and studies that have looked at how they vary between individuals and how they contribute to our understanding of what's different between us and even our last common ancestors in terms of our nearest non-human primate relatives. And so this is just if you think about a standard repeat, like if we're thinking about a book again, you're saying the dog jumped over the fence. I'm just highlighting the word dog in red so you can see this is a repeated sequence. However, when you start looking at centromeric regions, this is what we're dealing with. It's no longer just a catchy red word that you see kind of enriched in one space. You see the same type of repeat, the head to tail orientation over and over again. And what I'm trying to use for this book metaphor, and sometimes this can be 100 pages. So if you're talking to me and you're like, what sequence lives in our centromeres? I would tell you, oh, it's this 2000 base pair repeat that's found in the head to tail, sometimes up to 10 million bases long. And we have no idea why our genome, at this point, is organized in this way. I often tell individuals I work with, it's almost like you got to a point of the genome when you're listening to a record and it just starts to skip over and over again. And we see this in centromeres in humans. This is not a human-specific feature. You can look in plants, you can look in insects, you can look around all of life, and we find that this is a largely a trend in most I wouldn't say all, but there's always exceptions to the rule, of course, in yeast and species, but typically in life, we find that their centromeres are defined by these long chains of tandem repeats. And so when we entered into this new era of complete genomics, our team, where we met each other for the very first time here on campus, this was at the hay barn. You can maybe see Cindy in the middle of all of our T2T group, and my two kids kind of T2Ters in training when we first celebrated the release back in 2022. In red, you're seeing all of those sequences that our team was able to pull in to these new maps. And it really was because of these nanopore reads, where we had super long reads, where we could start to put the puzzle together instead of these shorter reads, which we had to use. This is where I get to embarrass Dave. So I get to put his slide on. As well, just to say, this was crediting Santa Cruz because once again, we were forefront of long read sequencing technologies. This is taken from one of Dave's notebooks back in 1989, where he had conceptualized the first nanopore technology of actually passing the DNA through a pore and taking reading. But when we started this work, we were able to read DNA up to millions of bases and generate data which we could see as ultra long. And we can now even do this in my lab. We're running a Seq Tech Center where we have like mini-ions, which I'm showing can be held in your hand, or you can use more Prometheans. And this is just a video showing how the DNA can actually be fed through the pore itself in order to read out the base signal. So these reads are really important because, as I was telling you before, lots of their gapped regions had these tandem repeats. And if the reads were greater than, let's say, 200,000 bases, we could almost bridge and completely span all of these particular gaps, which was critical to the success of the project. We also were able to close all of those duplicated regions. Here I'm showing you in red all the new genes that were introduced in our map that had not been seen before in our previous map of the Human Genome Project. This is really crediting my colleagues and collaborators, Evan Eichler and Mitchell Volger, who led our segmental duplication team as part of the T2T effort. And this is important because if you start to think about particular genes of interest, like the lipoprotein A, we know that this particular gene is an increased risk of cardiovascular disease. You can see maybe in the schematic that I'm showing you here in red, once again, to draw your attention, there are certain patterns of these genes that we know that if you have these particular domains, you have an elevated risk of heart disease. If we look at our chromosomes, so I said, okay, some of them have what we call acrocentric, or they're very short arms. We have 5 of them in the human genome. And until the T2T consortium, we really had no idea what existed in this part of our genome. And here I'm just trying to show you a blow up of that particular part of our map. And I'm highlighting for you in green and pink the different genes that we had no idea that we're there. And now we have a more complete map. We can see if they're being expressed, if they're creating an RNA product. We can tell if they are being methylated or if they're being regulated epigenetically. And then all of these different color blocks that I'm showing you, these are all the rich repeat classes that we had identified for the first time and began to draw out kind of the organization of these arms. Below, you're seeing this giant heat map. And what I'm trying to highlight here is mainly just a huge high five to everyone on our team, because these short arms are near identical to one another. So if you took a sliding window, right, and you were to go through each of the arms, we're looking at a median interchromosomal identity of 98.7%. So actually putting these assemblies together correctly was a Herculean effort. And so this is something that I just wanted to highlight here as well. Why care about short arms? We haven't had them for 20 years. Why do we need them now? I guess I would argue that we have known for 20 years that these short arms are involved in some of the most common structural human abnormalities seen in general human populations. One in particular we're going to focus on is the Robertsonian translocation, or ROBS. We know they have an incident rate of 1 in 1,000 births. However, I told you there are 5 of these acrocentric chromosomes. However, we don't see all 5 equally involved in these types of chromosome rearrangements. Rather, we commonly see 13-14, 21-14, and 21-21. So now, instead of showing you those acrocentric short arms kind of through a sliding window to try to prove to you they're very similar at that sequence level, I can start to pull the arms out so you can start to see each one of them and their satellites, banding patterns, as well as their coding and non-coding sequences as well. And what we found when we began to study the different arms is that we would find similar blocks of sequences on those particular Robertsonian associated parts of the chromosomes, chromosome 13, 14, and 21. What I'm showing you in yellow is, of course, the arrow. You can see that they're in different orientations based if you're on 13, 14, or 21. And this was really exciting to our team. I am eager to update my slides because now we have 2 different working groups that came out of this particular effort. One who's been studying translocations of Robertsonians, another one that's been expanding in our understanding of acrocentrics more generally. Now, my team was leading the first genomic maps of human pericentromeres and centromeres. I always like to emphasize how I never thought in my entire life I would stand in front of an audience and show this picture. It always seemed like it was a dream to be able to see any centromeric region, period, and not to have these maps now that we can have, not for one genome, but now thousands of genomes, and begin to study this rich amount of information. Now, if you're just glancing at this, what you're probably noticing off at the beginning are there's some really big colors that are catching your eyes. Maybe on chromosome 9, you're seeing this giant block of blue. That's a classical human satellite array. We know that one's 30 million bases long. It had not been characterized in any prior genome assembly effort. And if I were to walk you into it, it's a wild state of a genome where you can find it flip-flopping in orientation backwards and forwards, almost like it's making some type of origami shape. If you look at chromosome 1, your eyes may come into this dark purple. That's another amazingly important classical human satellite array, where we know that there are certain disorders in humans that have that particular chromosome go through misregulation. But what you're seeing maybe as a trend is that there's a red block on every single one of these particular centromeres. That's alpha satellite. That's the one that we've known in the community to be a really important site in your genome, because we always find the kinetochore, or those proteins that are responsible for proper chromosome segregation. They always go to those red parts of your centromeric regions. If we zoomed in, I will tell you that it's not all satellites, even though that's the joy of my life. There are genes that exist in these regions as well. Some of them, folks had kind of said, oh, we think there might be a gene there by other mapping techniques. But now we're even finding long non-coding RNAs that we're seeing expression from. We're starting to see all kinds of predicted regions as well. And so now it's a new kind of area to mine for new genetic information within and between these satellites within centromeric regions. Now, one thing I didn't tell you about this whole nanopore technology is that if you read the DNA through the pore, you can actually start to see not only the signal of the DNA bases itself, the A, T, C's, and G's, but you can begin to look at this, what sometimes we say the 5th base, or the modified sequences if they have a chemical modification on top of the DNA. And I'm trying to highlight this for you as being red if they're modified versus blue. And so now that we have these long sequences, and I can begin to add that other layer of information, do you have a modification or not? And we plot them on top of these really difficult centromere regions. We started to see these really cool trends. All of a sudden, we would see across the blue, which is this long chain of tandem repeats that I was highlighting before, you're seeing this kind of plateau appear. That's all of the methylation. Now, we expected there to be some methylation. What we did not expect to see is this very strange dip that was happening in the middle of all of these huge multi-million sized arrays. And when our teams leaned in and started to do some careful biochemical studies of what chromatin or what the proteins were that were sitting in these dips, we found that yes, there are important centromere proteins that are being marked at these locations within the dips. One in particular is centromere protein A, which we know from the community of individuals who care deeply about centromeres, that this is a fundamental mark of where kinetochore assembly or where the centromere is going to form. I often think people miss, I bring everyone's attention to those dips, but for me, it was so exciting to see what I call like more of that plateau or the mesa. That was very strange. So if I were to zoom out, you don't see that type of like straight up regularity of methylation in many parts of the genome. So it did signal to our team that there's something else going on, something special across the entirety of these arrays that we're currently trying to chase on and understand a bit more. Here I'm trying to break down the whole genome where you can start to see the size of the array, which is in light blue versus the ones that we don't see the kinetochore forming on. And you can instantly see there's something going on where the methylation is up as well as the size of the array. If we focus on one of these interesting cases, this is chromosome 21. It's often thought about in terms of chromosome biology because of trisomy 21, where we know we have a common transmission of 3 chromosomes, chromosome 21, we have a trisomy 21. And when we look at their centromeric region, once again, it looks very irregular in terms of its size and also the amount of methylation that it has across the span of this entire array. And I'm just trying to highlight that for you here. So these complete T2T assemblies have been revolutionizing the way that we do genomics. We've started working with individuals who want to study rare disease. So this has led us in a wild new group of studies thinking about how to use complete genomics for important clinical variants. We've been working now to launch several of these huge T2T projects with ruminants. We just celebrated our first non-human primate group of T2T papers. We have T2T frog, mouse. I was joking, we even have T2T corn. It's just name your favorite noun. We put T2T in front of it. But it's been a huge boom for trying to understand how our genomes evolve over time at the chromosome level. And we've been really expanding new cell biology. The big question, of course, that our team came up with is, okay, great, we have a new reference genome that's more complete, that we can start to see new biology that could be important for understanding human disease. But, you know, is it just one human reference genome? And is there a benefit of having more than one human reference to really understand the impact on human health? So this is another point where I get to step into a different role. I was the co-lead of the Telomere Consortium, and there was a lot of excitement at the time of the resources and new maps we were generating. And this was about the same time, 2019, when there was a call to action from NIH to launch a new equivalent of a human genome project. And this one was called the Human Pan-Genome Reference Consortium, where the goal was not to generate a single complete reference, but rather to do this to capture global genetic and genomic diversity, to at least reach 350 reference genomes around the world. So I'm bringing us back to this original image where I was showing you the human reference genome, where we now have the 70% from one individual that I had at the beginning of my slides, 30% and 19 other, but largely we've been organizing it as a single map. As I told you, maybe in the GPS model, your chromosome 1, position 1, chromosome 1, position 1,000. What we're doing now with the pan-genome is we're taking collections of these linear maps and we're organizing them into this new data structure. It's kind of more in the metaphor of thinking of a subway map, where now the parts of your genome that are shared with others, you can imagine as you're taking the same path, and where you differ, get on another track is where we can highlight this type of variation that we see that exists in human genome. And so this type of new structure where we can begin to study variation is really important. It allows us to study, it allows us to avoid genetic and genomic biases, which I'll talk about in a minute. So this is important for some really clinically relevant parts of our genome, like HLA typing, SMA loci. This is a leading cause of infant death, which we know is a complex region, which we don't typically have in any one genome well represented. And even the CYP2D6, which is responsible for 25% of clinically used drugs. That particular locus could be very different in my genome than your genome, and having multiple versions of genomes can allow us to study it more completely. So if we think about the T to T genome, which I just spent a lot of time walking through to make sure you could value it as much as I do, I'm now going to show it as this type of icon where it's kind of a finished one reference genome. This is quite different than if you were to go, let's say, to a hospital here in Santa Cruz or even up in the Bay, and were to do routine genome sequencing. Here, typically your physician might advise this for any certain type of genetic screening, and then you get a large number of sequence reads coming out from a DNA sequencer. If we think about this routine genome sequencing on the top, what you're looking at is not your whole chromosome, like those really maps that I just talked about, but rather small fragments of your genomes are being sequenced, and these are sequence reads. Now let's think about the T to T genome at the bottom, and we'll call that the reference genome. And now we can begin to map your sequence reads to the reference genome. And that's really fantastic because now we can go through and we can say, oh, I see at this particular spot in the reference genome, chromosome 1, position 1000, the reference genome has a G, but your genome happens to be a T, so now I can genotype and I can go through and call all these genetic variations. And this is something that we now do pretty routinely with reference genomes. We identify variants, we do routine genome sequencing, and we report these results to you in typical DNA tests. However, there's a problem, and that is if your genome contains information that's not present in the reference genome. Here I'm showing that in this type of schematic where the reference genome has a gap or a lack of information, but your genome has information. We call these large areas of the genome where we have a mismatch, or in other words, if you have insertion or deletion of information that's not shared that's greater than 50 bases, you'll hear this called structural variation or structural variant. And so now if we do the same type of exercise and we're mapping our sequence reads, what I'm wanting to highlight to you is that there's a section of reads from your genome that are not going to be mapped to the genome in a correct way to where we can call variants. We call this reference bias, and this is happening across a lot of our medical genomics fields right now. The pangenome tries to get around this. And the way that we're doing this is by taking that type of subway map or taking that type of collections of genomes. We can go through and do routine genome sequencing again, but this time the likelihood of having an individual that contains that type of information in that particular reference map is higher. So we can find a better mapping and improved mapping and better variant calls. Of course, it would be everyone's nightmares if I wrote to them and said, hey, the pangenome's out. Why don't you and your students download these 700 phased assemblies? I think we'd get a lot of no thanks because these are really big files. So the way that we've come across to do this in a more elegant way is that we identify the sections of your genomes that are shared. I'm highlighting that for you here. And so then you can generate this type of sequence graph where the nodes that I'm highlighting are the shared sequences and the edges are the adjacencies between those particular sequences. You can, in other words, take that information across hundreds of genomes and you can reformat it into this informative data structure. Each genome that's in our map has a path that we record, so we don't lose any information with this reformatting. So now if we think the reference genome, we go back to my little icon for station 13. Now I'm trying to lead you into thinking about it as being one of many, where we have several different reference genomes now that we've been collecting throughout the world. When we think about how to represent a human pangenome, this is how we're visualizing it. We actually have things called tube maps, where if you were to download any of our software, you can actually begin to see these types of things. You can see single nucleotide differences. You can begin to see things like duplications that we talked about, these paragraphs that are being changed, we can model. We can see insertions, deletions, as well as whole inversions that we know are taking place in the genome. So every change that can happen in your genome can be modeled by our team. So why is this resource so important? We really think about this as being a critical step in mapping new disease genes and predicting disease risk and diagnosing genetic disorders. And this is because it reduces this type of bias, improves the call, and it leads to more equitable genome analysis. So when we think about human genetics and genomics, we often ask who benefits. What I'm just trying to show here is a modified figure from Martin et al. In 2019. When we think about genome-wide association studies, these are ways for us to track associations with your genotype, phenotype, in this case, disease. We're seeing that there's a huge influx of this type of information over time. And if we think about the global population being projected here as that one set plot, and when we start to overlap population descriptors into this type of graph, what I'm showing you now is that we have a vast, overwhelmingly collected data for European descent, and we're missing a lot of information around from our global populations. And this has led to bias, not only in the mapping, but in representation of our genomic resources. And so this is, once again, why we're working so, I guess, diligently to ensure that we have global genetic representation. We did celebrate the first release of that subway map. I want to be a little humble. It was only 47 genomes. However, this was a very strong beginning in terms of the new sequence that we introduced. We had over 120 megabases of new sequences, 282 billion bases of resolved assemblies, and we were able to characterize and report over 20 million of these small variants. If we looked at the CYP2D6-7 genes, this is the part of your genome that's responsible for some of these, metabolism of clinically important drugs. What you can see is this is kind of a snapshot of real data from that particular locus. And I'm highlighting for you in color the start and end of all the genes, with the start being in green, going to the end being in blue. And we can start to trace the path for all the various genomes, and we can begin to call the structure that we see at those sites in a highly reliable way. Okay, I think I'm near the end, so hopefully I'm not going too long. I'm trying to make sure I'm on time. But here I am emphasizing that was just the draft. So this was back in May of 2023. And what we just released on Monday was our next improvement. We had over 270 genomes now that we've released into the public. There's been a huge amount of energy around those particular genomes, but we're all looking for that big announcement to happen. So pay attention, please mark your calendar for 2026, where we're making the first step forward into the public domain to say, this is a new genome that we're going to work with, and we're going to move away from these prior linear assemblies. So the Human Pangenome Project, of course, is a critical global resource. I have also taken a strong stance to our team to ensure that's developed by and ensure that we provide benefit to the broader global community. In doing so, we are a driver project for a big Genome Association for Genomics Health, or GA4GH, which we've been building federated alliances with international genomics partner now for over 2 years. The goal here is that we have shared resources, standards, cloud-based workflows, and in doing so, we've been facilitating a lot of education, outreach, and capacity building. This is me and my team. We were in New Zealand just a couple of summers ago doing a long read workshop there, building up reference genomes, teaching pangenomics. We still have members of the New Zealand team that we host here at Santa Cruz to continue that training. This is another image of going to Cape Town. These are members of my team actually having dinner with many students around the continent of Africa that come in for these H3A BioNet workshops. I myself went to one in 2024. My team went in 2023. And on Friday, we're hosting students once again to come to Santa Cruz to continue training here. We've also been working to build long read capacity centers in Africa. This is in Johannesburg. And once again, in South Africa, this is funds that I was able to recruit from Google Health. And we've been able to position their own Promethean, and I've set team members in now to do on-site training. This is members from the Johannesburg team who are generating their sequence data as well, and this is in collaboration with the Congo. As mentioned, we have been really trying to broaden capacity and outreach. I see this as not only a call to action from the federal government, but also from our company partners. So we have a number of different company partners now that are stepping up and trying to help us reach a broader group of pangenomics and reference genome partnerships and members. So with that, I'll end my talk. I started off presenting the human genome as being this double helix or a map. I think what I'd like for you to think about it now is being a collection of genomes of one humanity, where we can begin to study the variation that exists between our genomes. We can use this technology to start to develop this new, to minimize it, maybe Google Map again, but this time a new and improved tool. And this has to be open access. So once again, we can start to support new basic science, new clinical genetics, and once again, start to expand our understanding of human histories. And so I'll end with this. I really feel like I've been pushing my team to not only think about the Human Pangenome Project as being these important genetic variants that we've talked about today and how they're going to have impact in clinical genetics and science, but also that the legacy of our project is really on the benefit sharing models of how we can impact patients, clinicians, and researchers from around the world. And so with that, I will go ahead and end my talk and really highlight this amazing team that I'm able to work with. Along the top are all the different schools that are part of the Pangenome and the Telomere Project. On the bottom is our funding, as well as our company partnerships. So I'll be happy to take any questions. Thank you.
Thank you so much, Karen. Really amazing. We have time for questions, and I think Bernie gets his hand up.
Humans using one animal. How does the human genome compare with other animals? What are the biggest differences between the animals and humans?
Oh my goodness, well, animals is a very big term, but I will bring it into the non-human primates like bonobo and chimpanzee and gorilla, who we've now gotten a wonderful new insights into those particular genomes. And we know that even with our closest relatives, where we expect there to be a very small number of nucleotide differences that impact the protein coding parts of our genome, the actual number of chromosomes change. These repeat structures that I was focused on are completely turned over. And so we do know that there's a distinct genomic difference between even our genome and that of chimpanzee, which we've shared a last common ancestor with, on the order of 400 years ago. And so when we think about humans in particular, I think many people, including Benedict Payton, who's a faculty member up on campus, have been looking over these large number of complete genomes to do comparative genomics. And in doing so, we've been able to collectively, not Karen, Miga, have been identifying genes that are human specific. And some of those duplicated genes that I was highlighting before, on chromosome 1 in particular, if I could highlight to just one example, we're finding that there are human-specific genes that are involved in brain, morphology. We can look at them in terms of patients who have different brain disorders and things like that. And so there are absolutely studies like that. We're also looking at ancient DNA studies to see what could be different from us from Neanderthals and Denisovans. And those are other aspects of where we're identifying traits and variants that happen to be human specific. So it's a very big but an important question that can be tackled by many different ways: comparative genomic, ancient DNA, and even looking across humans. Thank you.
What has been the impact on clinical trials, and what is the FDA's acceptance of DNA in conjunction with a clinical trial?
I'm going to interpret your question in terms of the pangenome and these new resources coming out. Is that correct, or just in general?
I'm particularly interested in hearing some specific drugs or some specific diseases that have benefited by the work you talked about.
Well, I will say that for an FDA kind of going into the clinical stages of clinical trials, these are multi-years, and they typically have been based so far on the original Human Genome Project. The T2T genome just was released in 2022, so we haven't seen many uses of that in an FDA-approved clinical trial. And so that has been, perhaps from the perspective of the pangenome work and the T2T work, a barrier to entry, because a lot of people are still stuck on these older genome projects. Now, almost everything that has the word gene or genomics in it has been benefiting in clinical trial space from the use of HG38 and HG19. That's an immense list. So anytime you have any gene that's being focused on that has already gone through some type of animal-based research or cell line-based manipulation before it went to clinical trial that benefited from the genome. To get us to bring in those new clinical researchers in a more immediate way, one of the paths in our subway graph is the HG19. And so we're hoping that will be an early entry into trying to pull more of this clinical genome and FDA support into using the pan-genome structure, because you're not changing that original coordinate map. So maybe they don't have to change some of their restrictions and start to pull into what we call HG38++, where we're kind of adding on to those resources. And so it's a poor way to say we don't have an exact plan yet, but that's how we're trying to move more into the clinical space. It's a really hard space for people who are on the innovative part. I'm sorry, I don't know if that's me or it's a really hard place because we're on the cutting edge of a lot of genomic research, and it usually takes a long time for that to reach the clinical genomic space.
Yeah.
Speaking of the clinical space, when I have my annual physical, I go over to the little local clinic and part with maybe a few milliliters of blood. Next day, that sometimes even same day, I'll get a huge after visit summary of everything they've learned from my blood. Sooner or later, I think that there's going to be human genome sequencing available to doctors in the same sense. So you give a saliva sample, and next day you come back with a huge read. I wonder what you imagine the first sorts of clinical applications might be.
I think we're already starting to model some of this now. And once again, it's not the FDA and clinical space. I want to acknowledge the last question, but we're thinking if you turn 18, so now I can be properly consented. And I say, I, Karen Miga, I'm a healthy person, not 18. I want to have my genome sequence. And they could build a personalized reference genome for me that the doctors, could keep in my medical files. And then if I go in and I'm doing routine care and they want to take a cheek swab or saliva, they're not going to be able to build a resource like this. But they would have on file this really Karen Miga specific map. And they could instantly go in and say, oh, you're perfectly fine. Or, oh, look, you're starting to have these mutations that take place in your genome. And why we care is in cases like cancer, where we know that there are these advancing signatures that could happen in our genome that could lead to targeted therapy. Why we care is if all of a sudden it's not just my genome, but my husband and his family and everyone's genome, and we want to look at a Miga personalized family pan-genome. So we now are doing pedigrees of these pan-genomes to see if we can track the transmission and find de novo variants or where a change is happening, that we can watch it through the transmission of someone's pedigree. And I feel like that's really where we're going to start to see some of these more immediate changes. My colleagues are in clinics right now. This is their day-to-day job. They work with patients. They lament your statement of saying, hey, we have someone coming in and we're going to do this one test and we'll do another test and we'll do another test. And it's almost like you're trying to figure something out, and it can be an odyssey of many years and too much money and lots of labs that are involved. And so they are pushing right now to do kind of a one test lab. And I think that there's a lot of things that we can learn about someone's genome with a one sampling for rare disease. And that's being modeled at different sites and different clinics around the world right now. I know in Boston, in Seattle, I don't think here yet, but I do know that these are exciting times to start thinking about how to do this right.
I mean, the huge advances in artificial intelligence, what that do is part of your and you build a project to see how you can best use that to pull the information. Because right now you assume strongly that a negative, no identified deviation, but clearly there's a spread in general. There are positive things as well that we can learn. So I'm just asking very broadly, how do you extract more information than that and just be before to the ability.
Absolutely. So it's a really important point. Like we aren't doing the disease analysis. These are outside. We're just building the map. So like Google Maps, they're putting the annotation on, and the annotation, of course, is a probability of likelihood given the genome that you're in and how the different episodes interactions. But we do know there are particular sites that are more likely to be associated with disease. With that being said, AI has just been a boom at most of our collaborations and sites. When we work with biobanks, when we're starting to work, this is how most individuals are either taking an AI approach to learn more about the literature to bring that in, to learn more about all the different data from phenotype to genotype, because now we have this recording more and more of genotypes and medically associated data sets. And so I think that's to your primary question. I don't know what's is that me?
I don't think it's you. I think somebody's hearing aid is, so somebody that thinks they're closest to where that sound is coming from, I don't think it's coming from here. If you don't know what I'm talking about, I'm sorry, and I guess it's not bothering most people, but some of us are hearing this high pitch, but I'm pretty sure somebody's got their hearing aid turned up so loud that when she speaks, it's causing it's overloading. So if you think you're near it, maybe you could poke somebody and tell them to turn down their hearing aid.
Yeah, I can speak away from.
I don't think it's, yeah, I don't think it was coming from here because I was trying to locate it. Like, why don't you continue? Sorry.
Yeah, but the AI is everywhere. So we use it for polishing to get the highest quality possible. We pilot all the data and we learn from it to make sure we trust that base. We use AI for predicting gene models now. We use AI for doing there's a protein sit here, so it's really infused in basic science. But since your heart rate, the statement I understood was more clinical science, that's where I'm seeing it most active is in these large genotype to phenotype biobanking initiatives, which is exciting.
And I was wondering, what proportion of the genes do you know the function or how the function?
There's roughly 20,000 genes in the human genome, and they're always coming up on different lists associated with functions. It would be hard for me to say that this is a subset of genes which have no function at this point. It's almost like if you were to go into any one of our databases and you find this particular like link RNA, you'll see like 6 papers where it was brought up in some type of screen or something like that. So I don't mean to give you a wishy-washy answer, but I don't know what component of genes don't have a function. I will say that you can knock out a ton of these genes, like blow them away, and you won't kill the organism, right? So the cell line will survive. So when you start thinking about critical function for like life, that's where I think a lot of really creative people have thought, how can we minimize the human genome to like the bare minimum set of genes for life in a certain situation? And, of course, the second you move it to some other medium, we die. But this has been a really fun project of trying to watch the synthetic genome community to try to identify the minimal functional units of life, which is an exciting space. But when you just say function, I think a lot of these big consortium got in a lot of trouble because they're like, everything's functional in the genome. They're just finding transcripts coming from everywhere. And it's really
more of a specific function.
Yeah, well, I think that we are now understanding that the genome is a combination of genes. And I think this was kind of echoed in the last comment as well, that many genes are working together. And so sometimes there's even redundancy in the system. And so I don't know and sometimes they have multiple jobs. And so I think that there's a moment of humility and being humble on stage, that we have a long way to go to understand all of the direct A to B functions of the genes. And so what we do now is we just build the database and we have the genes and we just list for anyone who's interested, all the different papers, all of the different studies that have shown some element of function for that particular gene. And most of our gene annotations have lists like this. And so that's unfortunately the best answer to the question. But great question. And hopefully if you invite someone back in 5 years with AI and everything, I'll be like, this is exactly it. You can go to this website and look it up, because I think that there is a bit of synthesis that needs to happen.
So one more question.
So I wonder how NIH funding situation is affecting you.
I know I had a slide. This slide, I think it's something very earlier. Yeah, so it's been super stressful. Everything I told you, this is an old slide. I think it was like a couple weeks old. Things have changed so much every single week. I just had a call with my program director on Wednesday, and he gave me something bad's gonna happen, pay attention to the news next week. I'm like, no, you can't say that. But I mean, all of this call to action was through NHGRI. NHGRI has been almost completely gutted in terms of the director, the administrative staff, all the people who are doing grant management, the history, the policy teams, the communication team. Eric Green, we worked together on some of the slide deck for public communication of the Pangenome. He was let go. The person underneath him, Vance, was let go. We now have a new interim director, but they're planning to restructure that whole department of NHGRI. There's been a huge amount of restructuring in terms of the research grants on campus. It's impacted my lab because I do take in a number of students from the RMI program, which is really meant to help advance some of the training for researchers who come from diverse backgrounds, and they've roughly lost their funding. I always like to highlight this from my colleagues in NCD who have been really good about communicating some of this attack on biomedical research. I also like to highlight that the work that I was doing in South Africa, I was at a meeting in Durban presenting on this when I got an email saying you can no longer spend money in South Africa. You know, you can, and I'm like, how am I gonna get home? And it's kind of like this. It's like a constant stop and go, and every morning I wake up to see if our funding's been canceled. And I think that's the truth with most of the researchers. I think there's a real threat that in about, if these threats are real, that there's going to be a cut of 40% at NIH or even higher at NSF, that this will be devastating with biomedical research. And even if they turned the key and said everything's back to normal now, there's already been profound damage from the number of graduate students that we normally take into our campus. I mean, and to speak as clearly as I can, if you were to talk to me in November of 2024, I was actively recruiting and we were growing and we were vibrant. I closed my door to taking in any new students, and my funding runs out in 2026, even with a little smaller, sure. It's everything's down.
When you say restructuring, who is restructuring? Are they medical people or people like you or Robert Kenneth?
The restructuring that was originally proposed by Congress back in June of 2024, that was even before Trump was elected. That one was the ICs, so there's a set number of NIH ICs, and they were trying to optimize the ICs by restructuring them into different units. The most recent report that I'm aware of is going down from 27 to 5 of these ICs, so that's a very disruptive kind of like who's going to be there, who picks up the phone when you call about a grant, you know, who knows you. And this particular award is a special award. It's through a particular IC, and if the IC goes away and they no longer have that person fighting for you, that's where I think a lot of us are quite nervous. I think there's a lot of doom and gloom, and there's a lot of optimism, and so I've got this wide range of people I talk to every day. I don't want to lead you into some darkness of a conversation, but it is something that I think is incredibly impactful to the public and at large needs to be very aware of. So, thank you.
Well, it sounds like the Trump administration's way of dealing with the population explosion. So thank you so much.
Thank you. Thank you again.
And thanks for giving us some of this time.